Oh, okay. So, like, why I choose this topic for my presentation. So, like, as you know, like the biomedical, like why the biomedical is important for today's era. Like uh, the biomedical science is the fundamental building block of medical practice. Like without any deep knowledge, we cannot evaluate and diagnose like the illness of our body. So it's an important subject that's essential to improve the health of the world of the population. So then coming in recent days, the nanomaterials have received great attention because of their exclusive characteristics compared to the bulk equivalents. And within like the nanomaterials are very small size. That's the best advantage for the nanomaterials. And in recent years, nanovessel have received some attention due to this exclusive character. And some studies prove that earlier studies prove that like the nanomaterial could induce dangerous effect in living organisms. And a reason because of this, like we are going for a like the authors going for a reasonable explanation for this concern is that the conventional chemical approaches for nanomaterial synthesis require toxic oxidant or reductants, which are essential for nanoparticle formation and stabilization. So coming to this point, like currently if you go for this problem, then the solution for this, like currently the plasma technology is gaining great attention as a prominent green synthesis method for nanomaterials due to its distinguished properties when compared to solid, liquid, and gas phase synthesis approaches. Like furthermore, the combination of nanomaterials and the plasma in biomedical application demonstrate several strategies, effects, and better treatment. And coming to then come, yes, this one. No, no, I'm like using the laser point to point out. Okay. No, no, I'm just like using the laser point. So this is the figure. We can see we are why we are using plasma. And you can use biomedical application. The plasma you can use directly for plasma medicine sterilization. And for nanomaterials, in plasma you can use synthesis surface modification. And for that you can use nanomaterials to get like cancer therapy and drug delivery applications. And then coming to the introduction part, like what is the need of biomedical. like in As we know, in general biological study, the structure, function, and growth, origin, evolution, and distribution of living organism, biology is important because it's help us to understand how the living things are in our body. Then the technique in medicine, agriculture, and the biotechnology and many others areas of bio have brought improvements in the quality of life. And the biomedical research is important because is the first step forward the creation of new medicines and treatments that help manage different types of health condition and issues. Then without this research, the prevention and cure of disease would be practically impossible. And this general field of research includes many area of both life and physical science. Like as you can see in this figure, like there are several biomedical application if you go through the nanomaterial. So from this, like a drug delivery, wound healing, and bone registration, and so on. Then coming to the next one, I want to describe like some about like how the plasma is formed and what is the plasma. Then okay. 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 Can I say this one? Types of plasma. Okay, so here I'm going to describe about like types of plasma. So there are two types of plasma. One is thermal and another is non-thermal. So both non-thermal and thermal plasmas are usually initiated by high energy electrons excited by external sources such as like direct current and plus DC charge and radio frequency and electrons cytron resonance. So both thermal and non-thermal plasmas are sustained by two electrodes connected to an external power supply. The space between two electrodes is filled with one or more gases, atmospheric or low pressure, and the plasma is created when a voltage is between two electrode are placed. Then coming to the next one, like I am showing here, like what's the key difference between the thermal and non-thermal plasma. So coming to the thermal plasma, a thermal plasma is nearly fully ionized, and the presence of heat is more here, and the high temperature and ionization rate in thermal plasma facilitate applications such as welding, cutting, and plasma sparing, albeit limited to heat resistance material. And then coming to the non-thermal plasma, here non-thermal plasma has a small friction of ionization, and here the heat is not like a kind of absence, is the heat is very less, and the cold, like cold is same as the non-thermal ion, and gases in non-thermal ion plasma creates milder environment which is more suitable for biomaterials and biosystem that prone to the thermal damage. So these are the few application of non-thermal plasma, and these are the few application of thermal plasma. Then coming to the next slide, here I'm going to review some paper about the biomedical application in plasma. So the first one is, so in this review, I'm going to talk about how the low temperature plasma method is going to kill the uh, leukemia cell. So let me, like before going to this review, let me give a clear idea about like what is the leukemia cell. So the leukemia is a cancer of the white blood cells, but some leukemias start in the blood, other blood cell type also. So even though the leukemia in certain cases is curable through chemotherapy and radiation therapy or bone marrow transplantation, so some drugs and therapies may increases the short-term patient's survivability. So but still have high rates of long-term morbidity. The prognosis of leukemia depends on numerous factors, and there is no way to prevent this disease. As an alternative to traditional therapy that could result in detrimental side effects and increased burden to the patients and their families. So here, the authors propose to use a plasma pencil generate non-thermal gas, also known as the low temperature plasma, to to killing those leukemia cells present in the environment. So then, so the plasma that are generated by the plasma pencils, you can see here. So the oxygen species, and in this paper, the effect of LTP treatment on a single tissue culture cell line of leukemia cells has been investigated to determine the antitumor activity of LTP and the long-term effects of plasma-induced cell death. So here they use a pencil generates a plone or a jet of non-equilibrium plasma at atmospheric pressure and low biology tolerant temperature using the dielectric barrier discharge concept. So traditionally, the DBS employed two electrodes insulated by a dielectric material. So the dielectric barrier discharge plasma formation ensure because of applied voltage in the kilovolts range, and the frequency is uh, in the kilohertz range. And then coming to this figure, here they demonstrate that the cells are freely floating. You can say the cells are freely floating in the media. So so that the LTP reactive species can interact with the cells, like to influence the viability of the cells. And coming to the next figure, here the Trepan blue expression assays showing the live versus dead cell after 3 minutes of exposure to plasma treatment sample at 12 hours. The cells that are dead absorb tryptophan blue dye while live cells do not. And like let me tell about something about the trypan blue exclusion. So allows for a direct identification and analysis of live, stain and dead cells in each population. And then coming to the next slides, so here these three data sets show the result of cell viability counts using the trypan blue exclusion is cells that are alive have an interact cell membrane and therefore do not let trypan blue and dye enter. The results, like you can see here like for first 10 minutes they are like the leukemia cells exposed to the plasma. For the first 10 minutes they are the killing of cell is like higher because of the high exposing treatment of plasma. And like after that it's very this three figure for like the cell viability determined at 12 hour for the first one, and the second one is for 36, and the third one is for 60 hour post plasma treatment. So the result, so you can see that like here the 10-minute treatment had approximately viable 95% of the cells, and we suggest that if the dose of the plasma is high enough, the effect can be immediate killing of cancer cells, while at lower doses there is no immediate cell killing. Then coming to the next one, in here the second image displays that they are using a jet plasma jet plasma process to for the T98G human brain cancer cells. So here the the jet system consisting mainly high power supply voltage and electrode dielectric here a normal jet plasma used for treatment cell in the presence of air gas flow. You can see the here they are using the gas in, and the plasma is generated here. The inner electrode is a type of injection needle. This one is injection needle, and uh, the gas is injected into the injection needle, and then the ejected through a 1 millimeter hole. And in the outer electrode via the porous alumina, cells were treated with jet plasma for 30 seconds and 30 second to 120 second. And then in the second figure is the source that the cell morphology analysis revealed that the grown pattern of the T980 cell was also affected by the jet plasma. So here the comparison is shown between the treated with plasma and untreated control. Here is the control one, and these four are for the they are treated with the plasma. And you can you can see here like for the 120, like compared to others, the 120 is the higher cell is the higher cell population after treatment of the plasma. Then coming to the next one, so here the first figures, the first one is shows that the percentage growth of T98G cells at 24 hour and 48 hour and 72 hour after treatment by MPT assay. The results are shown as the percentage of viability. And the last figure shows the effect of jet plasma of the colony forming capacity of T98G cells. Here the colony assay is as plasma exposure increases, the plasma treatments enhance cell death and inhibit the colony formalization on the T98 cell population. Like even the 30 and 60-second plasma exposure as a decline, colony survive up to 79% and 73%. And however, the significant drastic decline percentage of the was observed after exposure to 120 and 240 seconds, with the survival percentage of 48 and 13% respectively. So this shows that these treatments are inhibiting the colony. And then coming to the third review here, they are like they are manufactured a spray type atmosphere pressure plasma system with a newly designed arc free and arsenic plate to provide uniform AP plasma for biological research application. So it's a kind of torch spray system, and as shown in this figure, in this experiment they are using helium and oxygen have been used as a carrier gases for the stable and low temperature plasma process. So here, like coming to the second figure, it shows the effect of non-thermal atmospheric plasma on growth of HCT116 in the figure of A and B. And the LOBO, like there are three kind of cells, HCT116 and LOBO, and the SW480 is for E and F. So in HCT116 cells, result is a significant reduction of cell proliferation after 2 hour and 24 hour, and similar effects were observed in S448 and the lower cells from the figure C, D, E, and F. So, however, the S480 and lower cells shows more resistance to the plasma HCT116 cells with a lower reduction in a viability of observed. So coming to the next slide, here some of the result. Here the migration ratio was documented by the photography. This is the migration result at 0 and 24 hour and 48 hour. So you can see in the first figure And in the second figure, the plasma significantly inhibited the migration and progression of SCT116. And in here in the first figure, the cell migration assay, the figure in the figure A, the HCT116 cell were played at 12 12 well plate and grown to confluence in serum containing media, and the monomer was scratched with a pipette of tooth to evaluate the effect of plasma on the surface. And the wound healing was documented by the second one is the hitting wound healing was documented by photography at 0 and 24 and 48 hour, and the quantification of cell migration assay from the three independent experiments. Then coming to the next one, here they review on the clotted plasma selectively and the possibility of a paradigm shift in cancer therapy. So in the first figure you can see that, like, here is the determine the cold plasma effect in vivo, and they applied the closed plasma jet to a nude mice bearing scrabble, and they examined the mouse skin after cold plasma treatment and did not see any damages like after 2 or 5 minutes. And then one can see that the single plasma treatment leads to tumor ablation with neighboring tumors unaffected. You can see in the figure B, they are not affected that much like after the treatment. And then we found that like we can found that like tumors about 5 mm in diameter ablated after 2 minutes of single time plasma treatment. And whereas the larger tumors decrease in size, increasingly ablated tumors did not grow back, whereas the partially affected tumors started growing back a week after a treatment. And then coming to the second figure here, a strong like selectively effect was observed. The resulting is about 60 to 70% of SW900 cancer cells were detached from the plate is the zone treated with the plasma. You can see they are detached from the plane when when they are detached and when they are placed like they are affected by the like treated with the plasma, and they are the control one, they are still on the surface. And then coming to the next one here, in we can see the selective effect plasma treatment B16 cells treated with the cold plasma device for 0, 30, and 60 and 120 second. And for correlated analysis of the results, characteristic cells is viable, adaptive, and the last approach and dead. And then coming to the next figure, here they demonstrate the tumor growth of fluid plasma treatment effect on the growth of established tumor in a murine melon model. Here you can see like the difference between the control one and the other with treated with the plasma one. And then coming to the next figure here, the cold plasma treatment effect on the mass survival in a murine melanoma model. So here you can also see that the difference between the treated one, like how the plasma is like affected to the mice and the control one. And here is my conclusion. In the review one, they revealed that the leukemia samples treated with the low temperature plasma using the plasma pencils have less viable cell, which increases dosage of exposure. And in the review two, the plasma was not only inducing the death in T98 cells, but also significantly affecting the reproductive capacity of the cells and has a broad exposure dosage ranges of activity against the human T98G brain sensor. And in the review 3, they found that the non-thermal atmospheric plasma induced growth invasion and cell death in HCT116 colorectal cancer cells. And coming to the fourth one, the development of cold plasma treatment will cause a paradigm shift in a cancer therapy. And these are the some reference of my presentation. Thank you for your attention.
Okay. Just one question for you.
Okay.
Why there's a selectivity for cancer cell treatment
Sorry.
for your last paper?
Okay.
Why there's a selectivity for killing cancer cell instead of normal cell by using plasma treatment? What's the mechanism?
Like I didn't get your question. Can you repeat again, Professor? Your question.
What's the mechanism for this selectivity? Why normal no, for the fourth paper?
Mm-hmm.
Yeah. for cancer cell
Okay.
the case. Okay, why you have selectivity using plasma treatment?
Because like for cells, plasma, like we can say like if you go for any other process, uh,
So what species in the plasma causing the killing of the cells? You know,
yeah,
what species from the plasma leading to the death of the cancer cell? What's the mechanism? You
killing
know,
of the cells.
I just okay, okay,
I think yeah. Okay.
okay. Thanks, okay. What's next? Amisha, okay.
Uh,